currently, particularly with neuroblastoma, the way that we treat kids is if they have really high risk disease, they get a lot of very nonspecific and aggressive therapy. And in treating kids with cancer, you really need to think about not only just treating the cancer, but also minimizing the side effects of things, hopefully for decades of life that they have to live down the line. And so that's where I think understanding the metabolism or how these pediatric cancers use nutrients may provide an opportunity to develop new therapies that may minimize some of those very aggressive treatments that they currently get. The long-term goal is to first characterize the metabolism of neuroblastoma, and then to try to exploit the metabolic vulnerabilities of this cancer to design therapies to use in patients. But there's a lot of steps and a lot of foundational knowledge that we need to gain first.
So my research is focused on the development of multimodal optical technology for cancer research. The technology goal here is to develop portable multiparametric microscope that can help us to measure several metabolic parameters simultaneously on small animals in vivo, so we can have a comprehensive understanding of tumor biology. We try to understand the radiation resistance mechanism for head and neck tumors. So we potentially want to understand why some of the aggressive head and neck tumors are not responding to their radiotherapy, and after that we can develop better radiotherapy for head and neck tumors. We also want to try to push our technology for translational studies. For example, we try to use our technology to help the doctors make the decisions for the treatment options. We may be able to provide a personalized radiotherapy for head and neck cancer patients and breast cancer patients.
So androgen receptor is a hormone-stimulated transcription factor and the main driving force for prostate cancer progression. So the standard treatment for prostate cancer is the androgen deprivation therapy, ADT. Unfortunately, a majority of prostate cancer patients develop resistance to ADT and transition to castration-resistant prostate cancer, CRPC. Although next generation of ADT can significantly prolong the survival of the CRPC patient, but unfortunately, the patient will develop treatment resistance, and treatment resistance remains an incurable disease. Therefore, my research interest is to understand the epigenetic and the transcription mechanism underlying the treatment-resistant CRPC.
The research in my lab is mainly focused on understanding the mechanism of breast cancer metastasis. For example, the breast cancer, they develop at the mammary site, but the tumor will travel to another organ. So this is caused like 90% of cancer patient-related deaths. So we want to know why the cancer become metastasis, and then we can develop the therapeutic to prevent the cancer metastasis. The neutrophil is supposed to kill the tumor cells because they're our defenses in our body. But in some cancer patients, this neutrophil instead, they can associate with the circulating tumor cells and help them to survive in the circulation and travel to the distant organ to form the metastasis. In this COBRA funding project, we particularly interested to understand how the tumor cells, they can change the metabolism of the neutrophil. We have the money, right, to hire the people to do experiment, to work on this project. Mentors are really important for our junior faculty, help us to guide our project progress, whether they go to the right direction, and also help us to write grant and to apply for another grant.
The CCM Imaging Core actually offers the high-end imaging capability, while the CCM Metabolomic Core provide instruments for my metabolomic experiments. COBRA funding actually provide both the hardware and the software to drive the success of my COBRA project.
I think this COBRA grant helped me a lot in many perspectives. They helped me a lot to develop my cancer biology questions. From the mentorship, several senior professors helped me to develop my research program and their grant applications and student mentoring and people management.
working both in the clinic and also in the research laboratory. I wear several different hats, and the only way that that works is through support through an institution and then also support through grants like the COBRA grant. And so for me, it's been enormously impactful in enhancing the folks that I get to work with, the tools I get to use, and I think the impact that this research is going to achieve over the next several years.